We're now looking to our next speaker, Molly Shoichet. And she's an expert on the study of polymers for regeneration. She holds the Canada Research Chair in Tissue Engineering and is professor at the University of Toronto. She earned her Bachelor of Science degree in chemistry from MIT, the way nearly everybody else did who's spoken today, and a PhD from University of Massachusetts at Amherst. She's published over 340 papers, and her lab holds numerous patents for drug delivery and scaffold design. Her title of her paper is Polymeric Nanoparticles for Targeted Delivery in Cancer. Thank you.
Well, you know, I think like many of you, I was wondering why I was invited, and so now I know. I just had to get a degree from MIT. But thank you very much for the invitation to be here. And do I control the slides, or do you turn it on? Okay, so next. Okay, so I couldn't resist, you know, just listening to all the different types of talks here, and really my talk is going to be on one subject, which is going to be on the polymeric nanoparticles. But I just couldn't resist and just tell you that my lab does a lot of work in regenerative medicine and tissue engineering, and actually just listening to the last talk, we spend a significant amount of time thinking about the central nervous system and regenerative strategies in the central nervous system. But really, we're rooted in the polymers and really thinking about, I always think about our lab as doing biology-inspired polymer design. So thinking about the medical problem, the biological situation, trying to understand that sufficiently so that we can work backwards and design polymeric systems to overcome injury and disease. And so a lot of the lab actually works in tissue engineering, or this idea of creating three-dimensional structures to guide cells in 3D, of this view of engineering tissues. And the other side of the lab works in drug delivery. And the two sides really come together in the idea of perhaps stimulating endogenous stem cells to promote repair, where you deliver therapeutic proteins, for example, to do that. But then building on that, we've also been working in the area of cancer. And so that's really where most of the talk is going to be. But I just want to give you one more sort of introductory slide and say, in terms of actually spinal cord injury, we're looking at stem cell delivery to the spinal cord, either in this is hard to see, I know, but this is meant to be like a tubular construct, so having sort of like a nerve guidance channel for delivery. And we're also looking at less invasive strategies. We've also been interested in creating three-dimensional scaffolds, hydrogels, where we can guide where cells are going to grow through chemical patterns. So we do a lot of photochemistry and laser, femtosecond laser technology applied to creating chemical stimuli to guide where cells are going to grow. And it was really nice to see a talk on hyaluronan, although I guess it really wasn't a talk on hyaluronan, it was more a talk on how hyaluronan isn't good. But we've been looking at hyaluronan-based materials for local delivery to the spinal cord. And so this is a minimally invasive strategy to deliver drugs locally to the central nervous system, because as many of you know, there's the blood-brain barrier or the blood-spinal cord barrier. And so we can't use some of the same targeting strategies that we might use in cancer to get drugs delivered to the brain or spinal cord, because we can't take advantage of the vascular system for that.
Who said it?
Who said it? Well, most drugs won't cross the blood-brain barrier.
If you have inflammation, you have very nice CPR effect into the brain.
Well, I think it's an ongoing challenge, but maybe we can debate that afterwards. So really what our lab has been focused on in terms of the nanoparticle technology, and the reason why I thought, given when I was invited to a nanotechnology conference, I thought this is what I should focus on, is creating these polymeric nanoparticles for targeted delivery in cancer. And the work I'll talk about is mainly done by the people listed here, and this has been a collaboration with a number of different labs. So now for the talk that I said I would give. So I don't have to introduce actually the EPR effect because it was wonderfully introduced already earlier today. But this is the effect that we want to take advantage of. And of course, as was already highlighted, you need the passive targeting, which is taking advantage of the hyperpermeabilized vasculature in order to get the nanoparticles into the cancer tissue. And then we've also been interested in what's called active targeting, where we've modified our nanoparticles with antibodies so that they would selectively bind to the cancerous cells and be internalized and deliver their drugs there. And so this is what I'll talk about today. So the idea then is that we've designed these polymers that will self-assemble in water to form these polymer nanobeads or nanospheres, and that's what's depicted in green. And I've shown here that we've put the drug on the inside, and this is the standard, is to put the drug on the inside. But I'll show you an example in a couple of slides where we've actually put the drug on the outside as well. Our targeting ligand here is a monoclonal antibody, and we've started to look at the Fab fragments as well as the way for targeting to the cancer cells. And so the squiggly lines are polyethylene glycol, and so they're incorporated into the amphiphilic structure of the polymer to provide for long circulation time. So in designing these polymers, we were able to take advantage of what had been shown and what was known in the literature and incorporate that knowledge into the design of these nanoparticles. And our focus has been primarily in breast cancer, and so the antibody is Herceptin. And what's kind of interesting about using Herceptin is that it has its own clinical therapeutic as well, therapeutic properties as well. And so while it's our targeting ligand, it has a dual function of targeting and therapeutic. Okay, and so, because this is such a broad audience, I didn't really want to put too much chemistry into my talk, but I wanted to put some chemistry into the talk. And so this is the polymer that we've designed. It's really comprised of 3 well-known units, just put together in a different way. So it's comprised of lactide or polylactide, trimethylene carbonate, and polyethylene glycol. And so this comprises the backbone, which is hydrophobic, and the polyethylene glycol is hydrophilic. And so that's where we get our amphiphilic properties with this polymer. But there's a couple of unique things that I just wanted to point out in terms of the structure. So one is that even though this is our hydrophobic backbone, it has this carboxylic acid group here. And so we can play with the pH of our system when we self-assemble these polymeric nanoparticles, and we can take advantage of that to tune the size and also the critical micellar concentration of the nanoparticles. And so this is something initially we designed into the polymer so that we could attach polyethylene glycol to it. But not all of these carboxylic acids are modified, and so it gives us really another tool to play with. The other thing I want to point out is that the end of the polyethylene glycol chain is terminated with a furan group, and this allows us just to do what's called Diels-Alder chemistry. If anybody remembers any of their chemistry, it's just what's considered a click-type chemistry, and it allows us to functionalize our nanoparticles quite easily. So in terms of the polymers, we've looked at their self-assembly in water. We'll actually dissolve the polymer in an organic solvent and then simply put it into a dialysis solution, an aqueous solution. And as water comes in, the polymer will self-assemble into nanoparticles, and we can look at its critical micelle concentration, which is, in this example, it's quite low. It's 10 to the negative 6. And we can also just do a TEM and we can visualize what these nanoparticles look like. So actually, in all of the work I'll be describing today, these polymers were really synthesized in the bulk with a stannous octanoate initiating system, but more recently, we've synthesized these polymers under living free radical conditions or ring opening conditions, so we can make much more monodisperse polymers. And the reason that's important is because we can then make more monodisperse polymeric beads, and that then becomes important in terms of controlled delivery. But just to show you a little bit more about these polymers, as I said, the polymers dissolved in an organic solvent, and by playing, we can add a small amount of borate buffer to this solution, and then, well, really, this is the aqueous solution, and then this is put into dialysis, and just against water, we get self-assembly. But by playing with the pH of the aqueous solution, we can control the size of the nanoparticles. And so in this very simple example, you can see visually just that this is clear and this is perhaps translucent, just because the size of the nanoparticles is getting progressively smaller. And so that's shown in data when we've looked at dynamic light scattering data. We can see that the hydrodynamic diameter of these nanoparticles changes according to the borate buffer. We've actually done other studies looking at the pH, and this is just the volume percent or concentration of the borate buffer. And so you can see you can get quite small nanoparticles, probably around 30 nanometers, and they can go up to quite large particles, which maybe we wouldn't call nanoparticles anymore because they're above 100 nanometers. So most of the data I'm going to show you is in vitro, but we have ongoing studies just looking at these nanoparticles without putting any targeting ligands on them and seeing their biodistribution in animals. And so in our first studies, we worked with this. Has a humanized HER2. It's an overexpressing HER2 cell line, and that just means it has the receptors for Herceptin. And we worked in a non-skid gamma chain knockout mice because we didn't want to have the immune system initially, because this is a humanized tumor cell line. And in our initial studies, we looked at subcutaneous tumors. So these are tumors just grown under the skin. And even in this study, what we did is we coupled an Alexa 680, so just a fluorescent tag to the nanoparticles, and we followed them over time. And you can see that in this study, this is their concentration in the blood, that very quickly the concentration in the blood drops. And so by 24 hours, we have less than 10% of the nanoparticles still circulating in the blood. So you might consider that that's probably sufficient time to accumulate in the tumor. But one of the things we realized about this model was that the subcutaneous tumor is not properly vascularized. And so in ongoing studies, we're looking at an orthotopic model where the tumor is grown in the mammary fat pad. And these results we're still collecting in terms of biodistribution, and so that is just looking at where the nanoparticles are in the different tissues and organs in the body, and then also looking at toxicity to the tumor itself. So, but I wanted to go on and tell you a little bit more about our in vitro studies looking at these polymeric nanoparticles and how we could look for targeting. So, as I mentioned to you previously, these polymers have that little furan group at the end, and we could take a antibody. In fact, we've taken small molecule drugs, we've taken peptides, and we've also taken antibodies, and we can selectively modify the antibody in the Fc region so as not to affect its bioactivity, and introduce a maleimide group onto that. And then the maleimide and the furan group just react very quickly, that you don't need any coupling agents, there's no side products. And it's a very simple and facile way to modify your nanoparticle with a targeting ligand. And it's all done in water, so you don't have to worry about destabilizing your nanoparticle or affecting the molecule that's already encapsulated, or in fact affecting the protein that you're attaching to the nanoparticles. So as I mentioned, we're interested in breast cancer and being focused on Herceptin. And so we could control the number of Herceptin molecules or anti-HER2 on our nanoparticles simply by reaction time. So you can see as over time you get increased numbers or increased density of antibodies per nanoparticle. And similarly, you can control the number of antibodies per nanoparticle just by varying the ratio of the micelle to the antibody in that reaction. And so these just give us a couple different ways of controlling the number of antibodies on our nanoparticle. Although I'll say we've really only put up to 10 antibodies, or maybe 11 antibodies on one nanoparticle. So these, these chains we envision, there's about perhaps 4,000 to 6,000 chains, and each of them has a furan group. And so you could imagine you could put a lot, potentially or theoretically, a number of different targeting groups on the outside. But probably because the antibodies are large, we haven't seen thousands of antibodies per nanoparticle. So what was important for us was once we put the antibodies on the nanoparticles to make sure that they were still bioactive, and this is what we did with flow cytometry. And what we did is we compared the binding efficiency of our nanoparticles with the anti-HER2 to 3 different cell types. The SKBR3 cells overexpress HER2, the MCF7s are low expressing, and the MDMB468 cells are non-expressing. And so whenever, as all of you know, whenever you do any cell biology, you have 10 controls for one sample. And so the one sample is this pink bar, where these are our anti-HER2 immunonanoparticles binding selectively with the SKBR3 cells, or perhaps not selectively because these were all done individually, but we see a much greater interaction and binding with the SKBR3 cells than we do with the other cell types. And then we had a lot of other nanoparticles, plain nanoparticles, particles just modified. The yellow bar is with an IgG1 kappa, so like a dummy antibody, demonstrating that it's a selective binding to those cells. And then the last part of the story then is really looking at a combination strategy of chemotherapeutic and antibody and looking at a co-delivery strategy. And so in this example, as I said right at the beginning, we can put our drugs inside our polymeric nanoparticles, but we can also put them on the outside using that same Diels-Alder chemistry. And so in this example, we have our polymer. We derivatized it with the antibody. Sorry, it's a little hard to see the red on blue. And then we also modified it with doxorubicin so that we have a co-delivery of doxorubicin and Herceptin to these SKBR3 cells. And our question was, could we see toxicity? And what's interesting about Herceptin, and this was also alluded to this morning in the talk by Professor Bernholtz, is that it on its own is not toxic to the cells, at least in vitro. But in combination with a chemotherapeutic, it is. So this is just the chemistry that we did, where we have our nanoparticles, we modified them with the antibody, and then we modified them with the doxorubicin. And here you can just see this is the doxorubicin that we chemically modified with this maleimide group. So I know it's kind of too hard to see this little red thing here, but that's our furan group, and that's coupled. And through a series of controls, basically where we quench this maleimide group prior to coupling, we can determine that this is a selective chemistry and that those covalent bonds have indeed been formed. And so then what we wanted to do was look at the toxicity. And we were interested in comparing the toxicity of our SKBR3 cells, which are the breast cancer cells that overexpress the HER2, to an endothelial cell line, mostly because we did envision this going through the blood and we wanted to see how these nanoparticles would interact with endothelial cells. So actually, if you compare just doxorubicin is highly toxic, as we all know, and we're just looking at really the inhibition of cell growth and using that as a proxicity for toxicity. And so with doxorubicin alone, it's highly toxic to both the cancer cells and the endothelial cells. when you put both doxorubicin and the Herceptin on the nanoparticles, this is where we started to see a selective toxicity to the breast cancer cells over the endothelial cells. And this was not due to the, we didn't see the same toxicity when we had just the doxorubicin delivered on the nanoparticles. And so this led us to believe that it was a receptor-mediated endocytosis that was causing the increased cytotoxicity. And I don't know, maybe the lights are too bright for you to see this, but what we did is we looked at a series of nanoparticles to try and better understand the mechanism. So with just doxorubicin, I'm going to start on this side, doxorubicin actually will go directly to the nucleus, and it's bright red, and so you can see that right here, the doxorubicin in the nucleus. If we just had our nanoparticles and we put a fluorescent tag on the nanoparticles, can you guys see this at all? Is it too, no, you can see it okay? Okay. Oh, look, it's brighter. Thank you. With our nanoparticles, we put a fluorescent tag on the nanoparticles. And so you can see with just the nanoparticles, they're not taken into the cells. So here we've stained the cells with a DAPI nuclear stain. So you can see the cell nuclei is blue. So, and we don't see anything around the membrane. Here, with our nanoparticles, so our nanoparticles, remember, are just comprised of thousands of strands of polymers, and the antibody is on one strand, and the fluorescent tag would be on another strand. And here you can see that we see the fluorescent green, it's due to the fluorescence on the polymeric nanoparticles. So this is one reason why we think that it's a receptor-mediated endocytosis. And then when we have the nanoparticle with the antibody and the doxorubicin, we see that taken up into the cell. And the reason why it's purple is because the doxorubicin is red, and that overlaps with the blue. And if we only have doxorubicin, we do see it also taken up in the cell, but it's not to the same fluorescence intensity. And so we see a greater intracellular accumulation of the combination in the cells versus just the nanoparticle of doxorubicin. Okay, so the sign says stop. So I won't go then into the mechanism, but we looked at the mechanism to look at the mechanism of apoptosis. And so we understand this further proves that it's a receptor-mediated endocytosis. So what I've shown you then is that we've made these polymeric nanoparticles. We've got some interesting compositions and coupling chemistries, and we've started to see selective cytotoxicity to cancer cells. We've done some in vivo proof of concept, and really that's ongoing, and we really have to do some more in vivo studies in order to advance this technology further. So I will just thank you for your attention. I want to thank the researchers in my lab that did the work and our funding. Thank you.
Questions? Please. Name and...
Kester, Penn State College of Medicine. Excellent talk. You say that the materials can be encapsulated within the nanopolymer matrix. What's your evidence for that compared with it's just coating the outside of the spherical polymer that you've created? And do you see, depending on what chemistries of the three different polymers you put together, hydrophobic versus hydrophilic molecules being better adapted to get into that core?
Okay, so thank you for that question. There were a couple of questions. One of the reasons actually we put doxorubicin on the outside and not the inside is because when we put it on the inside, we really didn't get that much encapsulated. And so the hydrophobicity, doxorubicin is more of the hydrophobic drugs. So currently we've been working with docetaxel, and docetaxel is more hydrophobic, and we get more of the docetaxel encapsulated within. But your first question was really, how do you know it's inside and not just absorbed perhaps to the outside? And so maybe I can tell you about a different study we've done with FRET pairs. So we've taken two fluorescent molecules, one activates the other, and so that's what a FRET pair is. So you see the emission spectrum only because the first one's activating the second one. And so we've taken these FRET pairs and we've encapsulated them in the nanoparticles, and we've looked at that excitation emission spectrum. And so they'll only give that emission spectrum if they're closely associated and they're inside the nanoparticles. So that's how we know that we're getting maybe not the drugs, but we're getting something inside the nanoparticles. And the reason why we did those studies was because we wanted to understand the stability of the nanoparticles over time. Because one of our questions was, are they falling apart? And so we know that they don't, that they are actually quite stable, even in serum, for at least 48 hours. So that's, I guess, how I can answer your questions. Thanks.
Any further questions? If not, thank you.
Thank you.